study was initiated as a result of data that was actually missing from our natural history database on tuberous sclerosis complex. So we have a natural history database that was first established back in 2006. So it's undergone some revamping over the years. And when I was doing some analysis internally for our organization, I noticed that about 60% of our participants did not have recorded genetic results on file, which can be a big issue since it's the top requested variable for our researchers to utilize when they're looking at clinical data. So this was really the spearheading moment for this project in order to fill the gap in our database. Of course, we could have gone the route of traditional sequencing, but given the lower cost of whole genome sequencing these days, as well as the extended amounts of data and just the next generation sequencing high power where we can have that higher resolution, identify some novel variants, we decided to pursue whole genome sequencing as our next step. So it's really where we initiated it from. And in the process of getting started, I decided to try and incorporate a way to give back to the participants that are giving us clinical data, as well as biological samples. So some of the participants in our database provide either blood, tissue, or cheek swab samples to us from which we can obtain DNA. And we utilized those to kickstart this pilot study, to sequence these specific samples. And as a result of this, we submitted for regulatory approval to return results back to participants if they would like to have them. So this is really valuable for certain families. They finally have an answer, or for family planning, either that family is considering having additional children, or some individuals with TSC who are milder and disease phenotype will actually go on to live what we call relatively normal life and want to have families of their own. So knowing their variant status helps them with family planning and potential things like in vitro fertilization. Our pilot study consisted of 20 samples, and this was the number chosen because we were graciously awarded some pilot funding by Julian and Janice Gangoli, and they've continued to fund some of this project so we can sequence more samples. But what we did was we submitted 20 samples for whole genome sequencing to the Translational Genomics Research Institute. So very research-based in nature. And what we found was that we had a pathogenic finding in either the TSC1 or the TSC2 gene in 12 out of 20 of the participants, so just over half. And then in an additional 7 of the 20 participants, we found novel variants. According to the research in TGen, they are very likely causative just due to the nature of the variant, including one which was very novel, which wouldn't have been detected via traditional sequencing, which was a large inversion. And for people who don't know what that means, I like to just visualize it. It's basically the DNA was cut at 2 points, taken out and flipped upside down and reinserted. So traditional sequencing doesn't always catch something like that. But as you can imagine, flipping DNA upside down might cause a problem for downstream protein function. So just to reiterate, 7 out of 20% had a novel variant or something like the inversion where we wouldn't have been able to capture it previously. And only one of our samples had no clear variant in either TSC1 or TSC2, which are the 2 genes known to be causative for tuberous sclerosis complex. But this was consistent with typical findings. Typically 10% of clinically diagnosed individuals via our clinical diagnostic criteria don't have an identifiable genetic cause, but it's still a significant finding for us out of the project because it begs further research into what led to the TSC diagnosis in this specific participant without that result. And then out of those initial 20 samples, we provided CLIA certified results back to participants in 13 individuals, so 65% so far. And just last week, we actually submitted 2 additional families, and we're utilizing GeneDx as our partner for that. We have not yet heard back from 5 individuals from the pilot about whether or not they would like the samples. And after those results came back, we partner with Kate Richardson, formerly Kate Mowry, who's a certified genetic counselor at University of Texas Medical Center in Houston, Texas. And she provides genetic counseling along with the results. So we're not just leaving our families hanging. They actually get educated about what this means for them and their family. And of interest, one of of the novel results was a potential mosaic case where, not all of the cells in the person have the variant. And I did some digging in our database, and this person only had one major clinical TSC feature noted in their medical record, and did not have epilepsy in their chart. So there's definitely some correlations that are going to start coming out of this pilot as we continue. And then to expand our work as we move forward from this pilot, we're hoping to sequence at least 500 patient samples by 2025. This is a minimum number that was calculated by some of our collaborators that are very educated in biostatistics and big data. And this is to be able to effectively analyze any correlations that we might have with our clinical data. And if we're successful in obtaining additional funding along the way, I expect that we'll continue this initiative past the 500.